Hi, my name is Ishir Rao, and I'm very proud to present today NovoMol, Recurrent Neural Network for Orally Bioavailable Drug Design. My work is in the field of computational drug discovery. Drug discovery is the first stage of the drug development pipeline in which drug candidates are identified and sent forward to clinical trials. However, the drug discovery process is extremely costly and time-intensive, with upward of 2.6 billion and more than a decade of research spent on average per drug. And a major contributing cause of this is the extremely high 90% failure rate of drug candidates seen in the drug discovery process, due to candidates lacking clinical efficacy, having high toxicity, or having non-drug-like properties. As a result, there's a clear need for a time and cost-efficient approach to drug discovery that limits this failure rate and allows researchers to quickly narrow down their chemical search space. One promising approach is in silico de novo drug design, which allows researchers to computationally generate novel molecules with desirable molecular properties from scratch. And this allows researchers to explore a broader chemical space and identify potentially novel or improved therapies in a cost and time-efficient manner. However, improvements in the accuracy and scalability of these in silico techniques are necessary before widespread commercial usage is viable. In order to address these issues, I propose NovoMol, which utilizes state-of-the-art deep learning algorithms to design novel small molecule inhibitors with favorable molecular properties, saving millions of dollars and years of research in the process. NovoMol uses a multi-step process that begins with collecting 480,000 orally bioavailable molecules in SMILE string notation. I specifically choose oral bioavailability since 80% of drugs are administered orally, and drugs that lack oral bioavailability tend to fail in the drug discovery process. Then, I train an LSTM-based encoder-decoder model to learn off of this data set and then design novel small organic molecules in this SMILE string notation. And this model designs these novel molecules character by character, and these molecules could be used in the future as drug candidates. However, in order to sort through all the generated molecules that our model creates, I used the quantitative estimate of drug likeness, which is a metric used to determine how favorable a drug's molecular properties are. Quantitative estimate of drug likeness, also known as QED, ranges from 0 to 1, with a higher QED value indicating more favorable properties. And in my research, I used two QED thresholds to track how my model is doing and its progress across training generations. First, a QED threshold of 0.37 indicates that the molecule would likely pass Lipinski's rule of 5, and greater than or equal to 0.586 indicates that the molecule is likely to have favorable orally bioavailable properties, which, as I mentioned earlier, is extremely important. Then, I take inspiration from the success of evolutionary algorithms in the past and use transfer learning to then retrain my model across 5 generations. And we find that across generations, the model creates improved designs for the generated molecules with more favorable properties, and which is seen through higher QED values. This can be seen here on the left-hand side, where I conducted principal component analysis on each of the generated molecules across all 5 generations. And what I found is that across generations, the model optimizes for a smaller and smaller, more optimal part of the chemical space. On the right-hand side, we can see that this is also reflected, where across generations, the percentage of molecules passing our 2 thresholds consistently increases, with 96% passing Lipinski's rule of 5 and 76% passing our oral bioavailability threshold. Here on the left-hand side, you can also see that before our evolutionary training method, there's a widespread in QED values of generated molecules, whereas afterward, there's a significant skew to the right, indicating that the model successfully learned how to design molecules with these favorable properties that is seen through a higher QED value. Finally, here on the right, you can see actually some of our top generated molecules in our 5th generation of our model. And at the bottom, you can see the percentage of molecules passing each of our thresholds across the generations. And while these results are extremely important and promising, it's important to remember that NovoMol's true application would be to design drugs for a specific drug target. As a result, I did just that. I applied NovoMol to design drugs for the oncoprotein PDGFR alpha, which is seen on the right-hand side here. And it's found in gastrointestinal stromal tumors along with leukemia. And this has been a significant drug target for researchers, as by inhibiting the function of this mutated oncoprotein and inhibiting the signaling of the PDGFR receptor, is proven to significantly increase survival rates in patients. And current oral cancer drugs, such as imatinib, are able to successfully inhibit the PDGFR alpha protein and have seen success in patients. However, these drugs also have several side effects which can impact the quality of life for patients. As a result, researchers are looking to design novel drugs with fewer side effects to target PDGFR alpha, which is what I apply NovoMol to do. In order to do this, I use NovoMol and retrain NovoMol on existing 415 existing inhibitors of the PDGFR alpha protein, utilizing the power of ligand-based drug design. And NovoMol is then able to design 311 potential drug candidates, with I virtually screen to calculate their individual binding affinities to the PDGFR alpha protein using the software Autodoc Vina, along with calculating the individual QED values of these generated molecules. And what we found is that 44% of our generated molecules had a better binding affinity than the current state-of-the-art drug imatinib on the PDGFR alpha receptor. And this indicates that less dosage of the drug would be required for the same intended effect on the tumors. And this is also important since it could indicate fewer side effects would be seen in patients due to a reduced dosage. And overall, this is really vital since it shows that NovoMol is able to design drugs that have a high binding affinity to a protein that it has zero prior biological knowledge for. So NovoMol does not know about anything about the protein or its specific task, but just from the molecular structures of existing drug candidates, it's able to design molecules with significantly more favorable properties and that are able to successfully bind to the protein. Additionally, 49.5% of our generated molecules passed Lipinski's rule of 5, and 16.4% generated passed our oral bioavailability threshold. And this is vital since not only does NovoMol design drugs to bind to this protein, but it also still focuses on designing drugs with favorable molecular properties that will ensure that these candidates have a higher likelihood of passing the FDA approval process. Identify 3 salient features of NovoMol. First, it is optimized to consistently design novel small molecule inhibitors with favorable molecular properties. And this will allow us to reduce the failure rate seen in the drug discovery process due to molecules with unfavorable properties. And due to these more favorable molecular characteristics, these drug candidates would likely pass FDA approval. Second, NovoMol is de novo, meaning it can tap into the complete chemical space rather than being restricted to existing molecular libraries. To put that into perspective, if the Earth represented the complete chemical space, we've only discovered a singular atom's worth of molecules. As a result, NovoMol being able to tap into the complete chemical space could allow it to identify novel and improved therapies that traditional methods could not discover. Finally, NovoMol is universal, since it can quickly be adapted to any protein target with any existing sequence ligands in just a matter of minutes, due to its generalized drug discovery structure that allows it to quickly pick up on important characteristics of drug ligands and pick up on those characteristics and apply them to design novel drug candidates for that specific drug target, even without any previous biological knowledge. And to carry on from this, in the future, NovoMol has truly limitless applications due to this generalized drug discovery structure. And some of the applications that I've already been looking at include for Alzheimer's disease, HIV and AIDS, and other types of cancer. We identified that NovoMol is an optimized de novo tool for drug discovery that has truly universal applications. As a result, NovoMol is a vital tool for the efficient design of potent drug candidates, which ensures that life-saving drugs can reach patients faster. Thank you so much for your time.